Hello everyone, myself Laxmi Bisoye and my teammates are Roshani Chaudhari and Namrata Chaudhari. We are the students of Shivajirao as Jondhle College of Pharmacy studying in final year B Pharmacy. Today I am going to explain you on the topic of herbal plants as immunomodulators. We have chosen this topic for our final year project under the guidance of Dr. Manisha Vite, Associate Professor. Content which are covered under this topic are immunity and its types, immunomodulation, history of immunology, methods for testing immunological factors, mechanism of immunostimulants and suppression, side effects of immunomodulator drugs, immunomodulation by medicinal plants, herbal plants as immunomodulator. Abstract. A tremendous increase in knowledge of herbal drugs constituents, their effects, and the side effects has occurred in recent years. Herbal immunomodulator is a substance which stimulates or suppresses the components of immune system, including both innate and adaptive immunity. The modulation of immune system by various medicinal plants products has become subject for scientific investigation currently worldwide. The present review summarizes some of the herbal plants with immunomodulatory action and also to provide insight into the future research in this area. Now, introduction. Modulation of immune function using medicinal plants and their products as a possible therapeutic measure has become an accepted therapeutic approach. Plants and minerals have been used since ancient times for the treatment of many ailments and diseases. It is now being recognized that immunomodulation of immune response could provide an alternative to conventional chemotherapy for a variety of disease conditions. Immunity is a homeostatic process, a series of delicately balanced, complex, multicellular, and physiologic mechanisms that allow an individual to distinguish foreign material from self and neutralize or eliminate the foreign matter. Now, the immune system. Immunity. Immunity is defined as body's ability to protect itself from an infectious disease. Types of immunity. Immunity are broadly classified into two types: innate immunity and acquired immunity. Acquired immunity is again classified into active immunity and passive immunity. Both immunities are again classified as natural and artificial immunity. This is the homeopathic stem cells, which is mainly classified into myeloid progenitor cells and lymphoid progenitor cells. These are the cells of innate and adaptive immunity. For example, eosinophil, basophil, neutrophil, and B cell and T cell. B cell and T cell comes under lymphoid progenitor cells, and eosinophil, basophil, neutrophil, monocytes, etc. This comes under myeloid progenitor cells. This is the mechanism of immunity. Now, immunomodulation. Development in clinical and experimental immunology strongly suggests that many infectious diseases and disorders arise because of stressful environmental conditions associated with suppression of immune system. It is evident that certain types of stress evoke physiological changes that influence susceptibility to infection and malignance. The ability to modify the immune response in animals and humans evolved from a desire to confer greater protection against infectious agents through a more complete understanding of the functioning of the immune system and of ways in which non-specific and specific immune mechanisms develop. Now, the history of immunology. Immunology is a science that examines the structure and function of the immune system. It originates from medicine and early studies on the cause of immunity to disease. The earliest known mention of immunity was during the plague of Athens in 430 BC. The yellow fever virus by Walter Reed viruses were confirmed as human pathogens in 1901 with the discovery of the yellow fever virus by Walter Reed. Methods for testing immunological factors. Many in vitro and in vivo methods are there of pharmaceutical screening. Of medicinal plants having immunomodulatory activity have been listed. In vitro methods include inhibition of histamine release from mast cell, mitogen-induced lymphocyte proliferation, inhibition of T-cell proliferation, chemoluminescence in macrophages, plaque-forming colony test in vitro, inhibition of dihydroorotate dehydrogenase. In vivo methods include spontaneous autoimmune disease in animals, acute systemic anaphylactic in rats, anti-anaphylactic activity, passive cutaneous anaphylaxis, Arthur's type immediate hypersensitivity, delay type hypersensitivity, reverse passive Arthur's reaction, adjuvant arthritis in rats, Collagen type induce arthritis in rats. Proteoglycans induce progressive polyarthritic in mice. Experimental autoimmune thyroiditis. Coxsackie virus B3 induce myocarditis. Porcine cardiac myosin induce autoimmune myocarditis in rats. Experimental allergic encephalomyelitis, acute graft versus host disease in rats. Influence on systemic lupus erythematosus like disorders in MRL IPR mice. Prevention of experimentally induced myasthenia gravis in rats. Glomerulonephritis induced by anti-basement membrane antibody in rats. Autoimmune uveitis in rat. Inhibition of allogenic transplant rejection. Now, immunostimulant. The term immunostimulation comprises a prophylactic or therapeutic concept which aims at the stimulation of our non-specific immune system. This implies primarily the non-antigen dependent stimulation of the function and efficacy of the granulocytes, macrophages, complement and natural killer cells. Mechanism of immunostimulation. Immunological defense is a complicated interplay between non-specific and specific cellular and humoral immune response stimulation and the suppression of the immunocompetent cells and the influence of endocrine and other mechanisms upon the immune system. Primary targets of immune stimulant are T or B lymphocytes or the complement system, and increase in phagocytes by macrophages and granulocytes plays a central role in immunostimulation. Activation of macrophages is probably important for the stimulating agents to remain in contact with the reactive cell. The second most important role in the stimulation of T lymphocytes, which can be achieved either directly or indirectly via macrophages. Now, immunosuppression. These agents could be used for control of pathological immune response in autoimmune disease, graft rejection, graft versus host disease, hypersensitivity, immune reaction, and immune pathology associated with infection. Out of the list, the maximum use of these agents have been prevention of graft rejection and treatment of autoimmune disease. Mechanism of immunosuppression. It suppresses rejection of transplanted organs, suppresses graft versus host disease, treats variety of condition which not completely understood, that is myasthenia gravis, systemic lupus erythematosus, etc. Selective immunosuppression for prevention of rheumatologic disease of the newborn. Side effects of immunomodulator drugs. There are various side effects are associated with the use of these drugs. For example, pulmonary toxicity, myelosuppression, increased risk of infection, hepatic fibrosis, lymphoma, nephrotoxicity, neurotoxicity, GI complaints, hypertension, hyperkalemia, hyperglycemia, renal dysfunction, tremor, diabetogenic, elevated LDL cholesterol, which is bad cholesterol.
First, ashwagandha, synonym withania root, asgandh, winter cherry, biological source. It consists of a dried root and stem bases of Withania somnifera, family Solanaceae. Geographical source Madhya Pradesh, Uttar Pradesh, Pakistan. Chemical constituent. First one is the alkaloids methionine, withferrin, anaferrin. Steroid lactones, withferrin A and methanolides. This is the structure of chemical constituents. Uses anti-stress, widely used in sex stimulant, sedative and hypnotic, hypotensive. Immunomodulatory action. Administration of an extract of the powder root of plant Withnia somnifera was found to stimulate immunological activity in bulb C mice. Treatment with a 5 dose of Withnia root extract 20 mg per dose per animal was found to enhance the total WBC count 17,125 cells per mm on the 10th day. Third one is a bone marrow cellularity 27 into 10 cells per femur as well as alpha hysterase. Positive self number 1800 upon 4000 also increased significantly after the administration of Withania extract. Treatment with Withania extra along with the antigen produced an enhancement in the circulating antibody tired and the number of plaque-forming cells in the spleen. Maximum number of PFC spleen cells was obtained on the fourth day. The Withania extract inhibited delay type hypersensitivity reaction in the mice. Administration of Withania extract also shows an enhancement in the phagocytic activity of peritoneal macrophages when compared to control in mice. Marketed preparation of ashwagandha. Product name ashwagandha capsule, immunity booster. Company name Dabur. Ingredient ashwagandha extract. Second is product name ashwagandha tablet. Company name Himalaya. Ingredient ashwagandha extract. Marketed preparation. Product name ashwagandha tablet. Second, Akasha Catechu. Synonyms: Katha Kach. Biological source: it consists of a dried aqueous extract prepared from hardwood and will and Acacia chundra wild. Family: Leguminosae. Geographical source: Madhya Pradesh, Maharashtra, Rajasthan. Chemical constituent: major chemical constituent of Acacia catechu are catechin, epicatechin gallate, procatechinin acid, alkaloid quercetin, and camphorol. Here is the chemical constituent of Acacia catechu. Uses: used in asthma, cough, bronchitis, colic, diarrhea, dysentery, boiled skins, effectation. Immunomodulatory action. Acacia catechu are shown an increase in the neutrophil adhesion to the nylon fiber, produce a significant increase in a phagocytic index, and the significant protection against cyclophosphamide induce neutropenia, indicating its effect on the cell-mediated immunity. On the other hand, Acacia catechu extract produce a significant increase in the serum immunoglobulin level, increase in the heme agglutination titer value, and decrease the humoral arm of immune system. From the above result, it was concluded that the aqueous extract of Acacia catechu has a significant effect on the both cell-mediated and humoral immunity. Marketed preparation. Product name Katha. Company name West Coast Pharmaceutical Work Limited. Main ingredient Katha powder, Acacia catechu hardwood, 100% W by W. Marketed preparation. Product name Westin 500. Company name Oasis Biotech. Main ingredient Scutellaria, Bacalensis, and Acacia.
Then marketed preparation. Product name Ginseng capsule. Company name Herb tonics. Ingredients contains panax ginseng root, vegetable cellulose capsules.
Fourth, garlic. This is a pictures of the garlic. Synonyms: garlic, allium. Biological source: it consists of bulbs of the plant known as Allium sativum Linn. Family: Liliaceae. Then geographical source: Central Asia, USA and India. Chemical constituents: 29% of carbohydrates, 56% of proteins, 0.1% of the fat, 0.06 to 0.1% of volatile oil. Volatile oil of the drug is active constituents and contains allyl-propyl disulfide, diallyl disulfide and allicin. This is a pictures of the chemical constituent. First one is a allicin and second one is a alanine. Then uses. First one is a carminative, second is a aphrodisiac, third one is a expectorant, fourth one is a stimulant, fifth one is a disinfectant, sixth one is a strong antioxidant effect, seven is a use as condimentum. Then immunomodulatory action. First one is a garlic, an important medicinal spice, displays a plethora of biological effects including immunomodulations. Second one, although some immunomodulatory proteins from garlic have been described, their identities are still unknown. Third, the present study was investigated to isolate immunomodulatory proteins from raw garlic and examine their effects.
on certain cells of the human system like lymphocytes, mast cells, and basophils in relation to mitogenicity and hypersensitivity. Fourth, three proteins components of approximately 13 kD like QR1, QR2 and QR3 in ratios of 7 raised to 28 raised to 1 were separated by Q spherorose chromatography of 30 kD ultrafiltered of raw garlic extract. Fifth, all the three proteins exhibited mitogenetic activity towards human peripheral blood lymphocytes, murine splenocytes, and thymocytes. Then marketed preparations. Product name is garlic extract capsule. Company name is Healthy Hay Foods. Then ingredients contains extract 1 raised to 13, alanine 2%, vegetable capsule shell, bulking agent, garlic salivum, and bulk dried sterilized extract. Fifth, Arjuna. This is pictures of Arjuna. Synonyms: Arjun bark, Arjun. Biological source: Arjuna consists of the diasteme bark of Terminali Arjuna. Family belongs to Combretaceae. Then geographical source: growth by the side of streams and very common in Chota Nagpur region. Chemical constituents: tannins, arjunic acid, arginolic acid, arginine, arginolone, and arginine. This is a picture of chemical constituents. Then uses. First, use has a cardiotonics. Second, it possesses diuretic and tonic properties. Third, it reduces the vaginal frequency. Fourth, it is also styptic, febrifugal, and antidiacetic. Then immunomodulatory actions. Terminally Arjuna bark powder is 400 mg per kg pound. Significantly reduces the formalin-induced the PAW edema. Second, at 24 hours but not carcinogen induce the PAW edema. It significantly increased the anti-SRBC antibody titer in the secondary phase of human response. Third, the same dose significantly reduced the durations of leaks and bites in both phases of formalin-induced the pain response and shows a significant increase in the tail. Flick latency at higher dose is 800 mg per kg pound. Fourth, these effects of T. Arjuna were antagonized by pretreatment with the Nexolon is 1 mg per kg IP. Fifth, these findings support the hypothesis that T. Arjuna has anti-inflammatory potential against some phylogenetic agents along with the some immunomodulatory activity and also has a antinooceptive action probably mediated versus central opioid receptor. Then marketed preparations. Product name is Arjuna Rest. Company name is Baidhanath. Ingredient contains the Terminali Arjuna, Vitis vinifera, honey, wood fordia, fruticosa, and Arista base.
Sixth drug is a guduchi. Here is the image of guduchi plant. Synonyms: Gulwell, Tinospora, Amrita. Biological source: these are the dried leaves and stem pieces of woody climber Tinospora cordifolia. Family: Menispermiaceae. Geographical source: India, Bihar, Konkan, Sri Lanka. Chemical constituents: Tinosporin, Tinosporica seed, Tinosporol, Gilonin, Berberin, Syringin, Cardiofoliocytes A, Tinosporocytes, Tinosporacytes. Here is the image of chemical constituents. Uses: use in general debility. Second one is use in pyrexia and skin disease. Third one is use in viral hepatitis. Fourth one is use in rheumatoid arthritis, jaundice and diabetes. Second one is prevention of fibrosis. Immunomodulatory action. An immunomodulatory protein in guduchi was purified from dry stem powder extract by an anion exchange chromatography Q Cepheros. Second one is immunomodulatory activity was assessed by lymphocyte proliferation and macrophages activation assay. The conformation of an immunomodulatory protein in Guduchi stem showing lymphoproliferative and macrophages activating properties reinforces and rationale of use of Guduchi preparation in several Ayurvedic medicine for immunomodulation. Here is the marketed preparation. The product name is Guduchi Veg Capsule. Company name Morphium Remedies. Ingredient contain Tenosporia cardifolia stem extract. Seventh drug is a Punarnova. Synonym hogweed. Biological source Bohorvia diffusa is a species of flowering plant in the 4 o'clock family. Family belongs to the Nectarinaceae. Geographical source Himalayan region. Chemical constituents B-sitosterol, A2-sitosterol, palmitic acid, ester of B-sitosterol, tetracosanoic, hexacosanoic, arachidic acid, uracilic acid. Here is the structure of chemical constituents. Uses. It is a cure disorder like intestinal colic, kidney disorder, cough, hemorrhoids, skin disease, alcoholism, insomnia, eye disease, asthma, and jaundice. Immunomodulatory action. The effect of purannovin on the immune system was studied using bird sea mice. Intraperitoneal administration of purannovin was found to enhance the total WBC count. Bone marrow cellularity and the number of alpha esterase positive cells were also increased by administration of purannovin. Purannovin enhanced proliferation of splenocytes, thrombocytes, and bone marrow cells both in the presence and absence of specific mitogens in vitro and in vivo. Marketed preparation. Product name Punarnava tablets. Company name Himalaya. Ingredient Punarnava Bohorvia diffusa. Root extract 250 mg. Eighth drug is a turmeric. Synonym Indian saffron, haldi, curcuma. Biological source. It is a dried and fresh rhizome of plant Curcuma long. Family Zingiberaceae. Geographical source West Pakistan, India, Malaysia and China. Chemical constituent curcumin 50 to 60%, essential oils bisbalone, dismethoxycurcumin. Here is the structure of curcumin. Uses helps to treat cancer. Second one is used in inflammation. Third one is a treat diabetes. Fourth one is a treat arthritis. Next one is a immunomodulatory action. Turmeric inhibits cell proliferation and cytokinin production by inhibiting NF-kappa B. Marketed preparation. Product name turmeric 95 30 vegetarian capsule. Company name Himalaya Drug Company. Ingredient curcumin, curcuminoids, turmeric powder extract. Ninth drug is amla. Synonym Imlica Indian gooseberry amla. Biological source. This consists of a dried as well as fresh fruit of plant Imlica officinalis girth. It is also called as a Phalanthus imlica lin. Family Euphorbiaceae. Geographical source India, Sri Lanka, Myanmar. Chemical constituent of amla. First one is a vitamin C, then gallic acid, ellagic acid, phenolbin, indole acetic acid. Here is the structure of chemical constituents. Uses of amla. First one is a diuretic, laxative, diabetes and dysentery, jaundice and coughs, antioxidant and antibacterial. Immunomodulatory action. Immune activation is an effective as well as protective approach against emerging infectious disease. E. officinalis has been reported to inhibit chromium-induced free radical production and it restores the antioxidant status back. Here is the marketed preparation of amla. First one is a amla juice. Product name amla juice. Company name Baidhanath Ayurved Bhavan PVT Ltd. Main ingredient vitamin C, natural immunity booster. Second one is a product name Chawanprash. Company name Dabur India Ltd. Main ingredient sugar syrup, water, amla pulp, honey, black pepper, sesame oil, numerous herb extract. Third one is a product name Amla tablets. Company name Dabur India Ltd. Main ingredient each tablet contain amla, Imlica officinalis 250 mg, excipient QS. 10th number drug ginger. Synonyms rhizome, gingiberis, gingiber. Biological source ginger consists of a dried rhizome of Gingiber officinale roski family Zingiberaceae. Geological source it is mainly cultivated in West Indies, Nigeria, Jamaica, India, Japan. Chemical constituent of ginger gingerols, volatile oil, gingeron, shawgols. Here is the chemical constituent of ginger. Uses first one is the antiemetic, second one is a plasmolytic, third one is a aromatic stimulant, fourth one is a carminative, fifth one is a flavoring agent, last one is a cough and cold asthma. Immunomodulatory action. Ginger stimulate or activate different cells of immune system to high against invading organism or misfunction. Ginger repress the high inflammation caused by hypersensitivity and activation of immune cells, thus manage the oxidative stress. Marketed preparation. Product name Sunti tablet. Company name Himalaya Drug Company. Main ingredient ginger extract. Second drug is a product name ginger capsule. Company name Healthy Hair Nutrition. Main ingredient ginger rhizome extract gingerols 5%. Third one is a product name ginger capsule. Company name Nutraleaf. Main ingredient ginger extract. Tulsi. Synonym sacred basil, holy basil. Biological source Tulsi consists of a fresh and dried leaves of Oseum sanctum lin, family Lamiaceae. Geographical source multi-branched annual plant found throughout India, cultivated in garden and also grow near temple. Chemical constituent of Tulsi. Tulsi contains volatile oil. The oil contains about 70% eugenol, 20% methyl eugenol, B. caryophyllene, carbacerol, cineol, linalool. Here is the chemical constituent of Tulsi. Uses antibacterial, analgesic, antipyretic, antihypertensive, stimulant, aromatic. Marketed preparation of Tulsi. First one is a product name Kafol syrup. Company name Charak Pharma Private Limited. Ingredient Haridra, Tulsi, Shunthi, Vasa, Bhibitki, Vasti Madhu. Second is product name Dibiocon. Company name Himalaya. Ingredients Gymnema, Tulsi, Indian Kino tree, Shilajit. Third one is a Sulin. Product name Sulin. Company name Hamdard. Ingredient dry extract आधा थोड़ा वसिका, तुलसी, dry extract licorice, dry extract Ocimum basilicum, menthol oil, cinnamon, sucrose. 12th one aloe. Synonym aloe kumari. Biological source aloe is a dried juice of leaves of Aloe barbadensis milo, known as a Quercus aloes or the Aloe Perry Baker known as Socotrine aloe or the aloe ferox miller, an hybrid of this species with aloe africana miller and aloe spicata baker, known as a Cape aloe family, belongs to the Liliaceae. Geographical source: Eastern and Southern Africa, India, Europe, Cape Colony, Northwest Himalayan region. Chemical constituent: contains 30% aloin, which is a mixture of three isomers: barbaloin, beta barbaloin, isobarbaloin. The region of aloe: it is the chemical constituent of aloe. Uses: strong purgative, antispasmodic, skin ulceration, irritation. Doses: act as a abortifacient. Ointment of aloe gel is used in sunburns, thermal burns. Immunomodulatory action: aloe vera has been shown to modulate the immune response. Macrophages have been shown to play the essential role as the first line of defense against invading pathogens. Marketed preparation: product name moisturizing aloe vera face wash, company name Himalaya, ingredient cucumber aloe. Second one is a product name aloe vera juice, company name Baidyanath, ingredient aloe vera juice. Third one is a product name aloe vera gel, company name Mama Earth, ingredient aloe vera extract, aqua, glycerin, sodium, PCA.
Conclusion: the use of various plant extracts and herbal fat additives in specific dose during the scheduled vaccination regimen may be helpful in obtaining higher protective antibody against different infections, including production and development of more effective cell-mediated immune response. For protection against various bacterial, viral, and other diseases, herbal formulation may be therefore recommended for use as positive immunomodulator. References: C.K. Kokate, A.P. Purohit, and S.B. Golhe, Book of Pharmacognosy by Nirali Prakashan, 52 edition. Halder S. Bharal and Medirata P.K. Kaur Sharma, K.K. Anti-inflammatory, immunomodulatory, and anti-nociceptive activity of terminally Arjuna rock spark powder in mice and rats. Clement F. Pramod S. N. Venkatesh Y.P. Identity of the immunomodulator proteins from garlic with the major garlic lecithins or agglutins. Immunopharmacol, which is edited in 2010 March. www.researchgate.net publication. We had referred this website also for our content which is required in our project, and various images which we have downloaded from Google. Acknowledgements: we are very thankful to Dr. Shivajirao S. Jondhle, our president, Dr. Geeta Khare, secretary, Dr. Nitin Mohire, our principal, for guiding us. Our aim is to provide you the quality knowledge. Hope you like this video. For more informative video, subscribe to our YouTube channel.